Now let's turn to the origin of multicellular organisms. Eukaryotes originated in a single cell, and they're called protists. Protists have now the mitochondria inside them. They're much bigger than any bacteria that came before. So a protist cell is about 10 times larger than a prokaryote. That'd be all the bacteria. So they're big enough to have the mitochondria living inside them. Now these protists, single-celled eukaryotes, originated about 1.5 billion years ago, and they can be classified into 3 broad groups: the slime molds, the algae, and the protozoa. From these, we get multicellular organisms. Advantages of multicellularity are a much, much larger size than can ever be achieved by a single cell with one fragile cell wall. You can have a division of labor between specialized cells, and a much greater potential for morphological or physical complexity. So here we have our single eukaryotic lineage, the protists, giving rise to these other 3 lineages. The slime molds then gave rise to the fungi, protozoa to animals, and the algae to the plants. Now, where do we get cells starting to live together? We recognize that as being fundamental to fungus, animals, and plants, but even in the bacteria, cells have the capacity to stick together. And in the case of bacteria, they're called colonies. So here are some colonial bacteria that can form quite elaborate shapes. These are all vast numbers of bacteria that are stuck together in this larger form. Now, these things can even sometimes appear as if they were a multicellular organism. They can move around together. And the key notion here is that it turns out multicellularity is not quite as difficult as one might imagine. That going from a single cell form to multicellularity really depends on how the surfaces of these cells stick together. And this is seen in these colonial bacteria. Those bacterial colonies that are able to move as a unit can move more efficiently if their cell walls stick really strongly together. So here's a number of different lineages of bacteria that are known to form colonies, and some are really quite sticky, almost like glue on the surface, and they can stay really firmly stuck together. With that larger structure, then they can move around. Now, we can, in fact, select for aspects of multicellularity in the lab. This is something that is known and really quite well studied. So yeast are single-celled eukaryotes, so they have the mitochondria inside them. Each cell is much bigger than that of a bacteria, but yeast is primarily a single-celled organism. Now, if you breed different lineages of yeast in the laboratory, and you do some artificial selection by taking those that tend to clump together, then they tend to fall out of the solution in the media that you're growing them in. And if you do this long enough, you can get them to produce quite large, what are called snowflakes, heavy snowflakes. And these are single-celled organisms. The surfaces stick together, and they're tightly forming a larger snowflake. And what's interesting is after this is done, and this doesn't take that long in laboratory experiments, you get these snowflakes that will then just start growing and growing, and they reach a certain size, and then it buds off. So in a sense, you have the beginning of a multicellular organism that gives rise to a multicellular descendant. What's even more striking is that it doesn't take that much of this kind of artificial selection in the lab to produce an important aspect of multicellular life. When you get this snowflake division, what happens is that there's certain cells here that die, and they actually are programmed genetically to die off. And so you actually have, in a way, a blueprint for a larger structure in the body caused by programmed cell death. And this is a form of division of labor, that these cells are willing to die so that the rest of the clone, the rest of the colony, can proliferate. This photograph shows a variety of colonial slime molds. Slime molds are single-celled eukaryotes that can merge to form quite large colonies containing over 20,000 cells. So what we see in these pictures are all colonies, and each different species has its typical form when it makes these colonies. The slime molds were the first eukaryotes to live on land. Evidence suggests that they colonized dry land about 1 billion years ago. We can look at modern slime molds and see some fascinating aspects of their life history that show how this origins of multicellularity could have taken place. So let's start with the asexual reproduction of what's called cellular slime mold. These release spores, and the spores then develop into amoeba. The amoeba then undergo meiosis, and they reproduce sexually with each other. So you have several of these coming together. There's sexual fusion. And then these amoeba that have now sexually fused again can merge, and they form together to make a colony. And then there's cell specialization. So some cells in this colony become stalks and make the fruiting bodies that give rise to the spores, and the whole cycle repeats itself. So let's watch some animations of this. First thing to keep in mind about cellular slime mold is that these are amoebas that remain separate cells. Even though they merge together, they're each one a separate cell, just like we have separate cells in our bodies. So we have these different amoebas. They've just undergone meiosis, sexual fusion, and now they're going to start aggregating. We'll see in this animation. So here we have separate cells. They're all single-celled eukaryotes. They can live perfectly well alone, but conditions favor certain times for them to merge to form these colonies. They come tightly together. And then what we know is that these can do this only with certain other cells. So in fact, they're genetically identical clones of each other, and they then have a surface protein that sticks very efficiently to another cell with exactly the same surface protein. So these are all genetically identical individuals that are merging together. And once they've merged, they can do some amazing things. So here's a slug of cellular slime mold that can now move along. Okay, so the cells are moving along in a way that gives movement to the overall structure. At this point, we see a clear division of labor. Some cells become stalks. All they do are a physical structure to hold up the fruiting bodies from which the spores can emerge. And other cells stay inside, guarding the remainder, and they remove bacteria from inside the slug. A second major group of slime molds are called acellular slime molds. These start out again with the fruiting bodies producing spores, but these are haploid spores that then undergo fertilization, and these amoebas fully merge. So they actually have one large cell wall outside now the diploid organism, which then undergoes rapid mitosis, proliferation of all these nuclei inside now a very large cell wall in the plasmodium. And this can aggregate again, or not aggregate, but it can now transform itself into different shapes and grow the stalks and fruiting bodies. So here we have an acellular slime mold that's proliferating. It looks a little like tributaries in the river stream. And these things grow outwards, and they can grow to be 3 feet across. So this is, again, a colony in the strict sense, in that each one of these can live an independent, separate life, but they undergo this mitosis occasionally and form huge, huge slime mold grossness. In tracking many of the most important aspects of life, an essential distinction is that between organisms that can produce their own food versus those that have to eat something else. First, let's look at what are called heterotrophs. These are organisms that obtain nutrients from other organisms. That is, they have to eat. And this includes in the multicellular organisms all of the fungus in the animals. Now, fungus include the molds, so penicillium is a mold. Mushrooms are fungus, as are shelf fungus. Now, fungus have to find something to eat. And the way they do this is actually through external digestive organs. So if you were to look at a moldy piece of food, if you were to look at the bottom of a mushroom where it's attached to the ground, you see some cottony stuff. And that cottony stuff is called mycelia and hyphae. Okay, these are very thin strands. And what these are doing is these are where they absorb nutrients. Okay? And amongst the living fungi, we have some that have one huge cell wall with lots of nuclei inside. That's like the acellular slime mold. And you have this ability then for these very cottony tendrils to make it inside a cell of a plant or another organism, rotting meat or anything, and absorb the nutrients through its surface. There are some forms of fungus that have separate cells in their mycelia, in their hyphae, and that's like the cellular slime mold. So this is the only time I make any distinction between cellular and acellular. The important point is that when we go from slime molds to fungus, these are now obligatory multicellular organisms. A fungus can only function as a multicellular organism. It may still, in some cases, not have a division between its cells, like the acellular slime molds, and sometimes it does. That's all we care about that. The key thing about fungus is that these are the first multicellular land organisms. They appeared about 1.3 billion years ago. And their mycelium, which are those digestive organs, that's what's key in turning dirt into humus. They can actually extract minerals from the soil, the actual physical soil, mix that in with organic material that they get from other dying organisms, and that actually turns out to be extremely good plant food. The mycelia, being on the surface and always being exposed to whatever is in the dirt, they're going to be exposed to an awful lot of bacteria. So the mycelia have a defense against bacterial infection, and we know those as antibiotics. That's where penicillin comes from, and erythromycin. Now, the largest living thing on Earth, as far as we know, is a huge honey mushroom that covers over 2,000 acres in eastern Oregon. Somewhere over here, there's one huge, long, genetically identical organism extending all those hyphae and mycelia all over the place. It's been alive for thousands of years, and underneath the surface, all those mycelia have been branching out and occasionally come up above the surface, and you see them as mushrooms. So that is the sum total of all we know about fungus that I'm going to discuss in this class. The key thing really is that they are multicellular. They have digestive organs on the outside of their hyphae, their mycelia, and so they're a great source of antibiotics. Now, what about the animals? The animals are also heterotrophic, and they too arose from protists, a separate lineage from the fungus. So we have ancestral protists, single-celled eukaryotes, that, as we'll see, gave rise to sponges, jellyfishes, worms of various sorts, mollusks, sea urchins, even us. And what we have in our earliest forms of protists that gave rise to the animals, here's an example, the paramecium. It's a protozoa, single-celled eukaryote, has its mitochondria in there, has its chromosomes, and it needs to eat. So here's something that's going around looking for food. It seems to have found a bit here. And the key quest of all animals' lives is to find something to eat. Organism this simple doesn't seem to have a good idea where its own mouth is. But it is going to find eventually something to eat, and that is its lifelong quest to get those nutrients in order to reproduce more and more single-celled organisms. Now, the earliest ancestor of the animals was likely this, a choanoflagellate. It's a colonial protist. So these, each one could live independent lives. They're eukaryotic cells like the protozoa. They have mitochondria in them. But they can form colonies. Just like the slime molds form colonies, so can the choanoflagellates. And it turns out that the ability to form a colony is really important to the biology of this very simple organism. And they produce 78 different proteins that promote cell adhesion, because how well these cells can stick together determines how well they can move around and manipulate their environment in their quest for food. In choanoflagellates, the colonies grow by cell division, so they actually just add new cells to an already pre-existing colony. And so this looks a lot like the embryo of an animal in its very, very, very earliest forms, with a number of different cells stuck together. Now, as we go from these colonial protists, we have a multicellular organism that we're now going to call an animal. And we may have a colony that sticks around in a ball. They can move around a little bit looking for where there might be more particulate food sources, and then grow out to form a hollow sphere. And at some point in here, certain cells become specialized to become the reproductive cells, and the rest become the body cells or the somatic cells. And this is a similar kind of division like we saw in the slime molds between the stalks of the fruiting body and those that end up producing the spores. At this point, there can also then be a large-scale reformation, as it were, of this colonial structure. It has an infolding, and the interior surface now can have very different kinds of function for each cell. These can handle, say, digestive juices. They can absorb the nutrients, while the outer layer of cells protects the body, provides some sort of protection against the environment. The earliest multicellular organisms that we recognize as being an animal is this thing. It's called Trichoplax. Trichoplax has different kinds of cells on its back than on its front. And then often it's like a little tiny pancake. It's about the size of a dime. And when it finds something to eat, it'll pooch itself up and surround the food item with these ventral cells, and they become then a temporary digestive chamber. So remember, animals are heterotrophs, and so their evolutionary history is dominated by the need for food. In modern classifications of animals, what's considered the most primitive or the earliest form of a true animal is the sponge. The sponge may seem inert, but if you actually look at what's going on at individual cells in a sponge, there's a lot happening. The sponge actually brings water in and forces it out. It has a central cavity, and water has to pass through the spongy surface. And as it does so, then there are these specialized cells that can obtain the food items. And these flagella here actually create a small current that draw the water in. And so they can get any kind of particulate food item that might exist. But there are other cells, again at this very small scale, have a very different function. These are structural. They hold these collar cells in place so that they can do their filtering. So we're going to look a lot more at animals in the next lecture. So we'll come back to this next time. The other main kind of organism is that which can produce its own organic nutrients. These are the autotrophs. So we've already seen in eubacteria that they're capable of photosynthesis. So are plants and algae. And they use photosynthesis in order to manufacture carbohydrates. They're autotrophic. All they need is sunshine to be able to produce organic matter. Now, let's think about these single-celled protists, the eukaryotes. They have the mitochondria. But somewhere along the line, some protists were coexisting with free-living cyanobacteria, reproducing on their own, and then a eukaryotic protist ingested but did not digest a cyanobacteria. And then this eventually became a chloroplast. These chloroplasts function exactly the same as cyanobacteria in their ability to produce organic molecules through photosynthesis. So we have now the algae. The protists until that point would just have been heterotrophic. They had to get their organic food source from somewhere else. The cyanobacteria were autotrophic. Now they're bringing the capacity for photosynthesis inside the algae. Algae are autotrophic because they have chloroplasts. So, if we look at the endosymbiotic origins of the eukaryotes, we have what were three free-living separate organisms: eubacteria, protists, and cyanobacteria. These all reproduced independently of each other. At some point, a protist absorbed but did not digest a eubacteria, and then later on absorbed but did not digest a cyanobacteria, and this gives rise to the algae. Aerobic abilities was absorbed then by the heterotrophs to become protists, and then autotrophic lifestyle can be absorbed by the protists to become autotrophic algae. Algae, free-living, single-celled organisms capable of photosynthesis, but they also form colonies. Brown algae merge to form kelp. Green algae and red algae in various ways form colonies of their own kind that we see as seaweed. These are colonial protists. From this, then, we have the origins of true multicellularity, the autotrophic plants. From algae, certain protists, algae colonized land, became plants, first as mosses, and then as they grew larger, they were able to have systems whereby they could transfer nutrients from the soil out to the stems. So you have the vascular plants, then woody trunks, different kinds of seed formation with the seed plants, which gave rise to the conifers, and then the flowering plants. So all these descend from the algae, those first autotrophic protists. We've seen the independent evolution of multicellularity in fungus, animals, and plants. And it appears that a key permissive factor for the origins of these multicellular organisms derives from increasing levels of oxygen. We've already seen the role of oxygen in preventing the continual reemergence of life from nothing. Once there was enough oxygen, it would destroy naked DNA and unprotected proteins. So we have, with the origins of photosynthesis, with the cyanobacteria, quite a remarkable increase in oxygen globally by about 2 billion years ago. What's only recently been recognized, however, is that there was another big increase in oxygen levels about 600 million years ago. As we'll see in a much later lecture, there was an extraordinary ice age that covered the entire planet. And this resulted in a lot of fertilizer getting into the oceans. This allowed photosynthesis now to boom with all the algae and the cyanobacteria in the oceans. Oxygen went up to yet a higher level at this 600 million year ago mark. And it's at that point that multicellular organisms could really take off, because then they could really get enough oxygen to be able to do all the physical processes necessary for multicellular life. And so about 600 million years ago, we see the most strikingly early multicellular animals. They have fancy names we don't need to know. But as oxygen continued to grow in our atmosphere, organisms could get larger and larger, even giving rise to things as big as dinosaurs, blue whales, and giant sequoias. So, what we've seen in this section first is endosymbiosis. And this explains the origins of mitochondria and chloroplasts, allowing anaerobic organisms to become aerobic and some heterotrophs to become autotrophs. We've also seen multicellularity, which evolves separately in fungus, animals, and plants, allowing a division of labor amongst the cells and much, much larger size.